All right, so some of you have asked for this description of metabolism. So I'm going to start in this first one and just run through metabolism of glucose. So here's glucose outside the membrane. Remember, just as a good reminder as you're studying for your test, right? If we want to get glucose into the cell, there has to be a glucose channel. And the only way that gets activated is if insulin over here binds to an insulin receptor, and then the insulin receptor opens up the channel so glucose comes into the cell. Once glucose is in the cell, phase 1, glycolysis. So during glycolysis, whole series of steps where glucose gets broken down into pyruvate. And remember, for our purposes, the important thing to remember about pyruvate is that we started with the 6 carbons in glucose. Pyruvate is a 3-carbon compound, right? Keep that off to the side, that we need to drive the rest of, or the aerobic steps of metabolism. Energetically, we had to spend 2 ATP on glycolysis, but we get out 4 ATP. So grand total in this phase of glycolysis, we end up with that pyruvate, which we actually end up with 2 of them, and then we end up with 2 ATP. And a little dotted line here. With this process, we could call glycolysis anaerobic respiration. The only other step that we would actually use if we call this anaerobic respiration is that the pyruvate, instead of heading down into the rest of anaerobic respiration, so that we could keep glycolysis going, we have to retrieve some of the things in pyruvate, and so the cell will go ahead and convert pyruvate into lactic acid. The only problem with lactic acid is that the cell that converts the lactic acid can't metabolize the lactic acid, so it builds up, is painful for the cell. Cell has to actually transport the lactic acid out of the cell. Lactic acid ends up going to the liver, and we'll catch up with that lactic acid later on, okay? So anaerobic respiration, when we have low oxygen environment, we end up with those 2 ATP and pyruvate converted into lactic acid. If there is enough oxygen, pyruvate gets transported down to the mitochondria. In the mitochondria, we see first step being the citric acid cycle, and then citric acid cycle is used to drive electron transport. Now, along the way, that pyruvate has to be converted, and what we see that conversion step take place, converts that pyruvate into acetyl-CoA. Worth mentioning at this point, acetyl-CoA, 2-carbon structure. So in order to get from that 3-carbon pyruvate to that 2-carbon acetyl-CoA, some of that carbon is going to end up being released as carbon dioxide. Acetyl-CoA is the intermediate then that drives the citric acid cycle. In terms of goals of citric acid cycle, so as long as there's oxygen, and we'll see where the oxygen ends up being used here in a second, as long as there's that oxygen present, acetyl-CoA, through a series of steps, we're going to rip off hydrogen. And so hydrogen is going to build up inside the mitochondria, and hydrogen is going to be used to drive ATP synthase. Real quick, back over to the citric acid cycle. So essentially those 2 carbons that were in that acetyl-CoA are eventually going to end up coming out during citric acid cycle. So all of that carbon in the original glucose ends up being metabolized and released as waste product by the body as carbon dioxide. So last steps here in electron transport, we're using those hydrogen ions, those protons, and as they pick up, as they gather in high concentration on one side of the membrane, they will transport through the ATP synthase, and they're providing the energy required to put that extra phosphate onto ADP to charge up ATP. Okay, and then the kind of last step, make sure I got enough room here on the screen, that oxygen, what we're using the oxygen for, is as hydrogen comes down, picture that eventually some of this hydrogen would want to build up on this opposite side of the membrane, and it would ruin the concentration gradient. So we take some of that oxygen, some of that hydrogen, pair it together, and we make water. So as a real quick reminder, what's the importance of oxygen? Well, if that oxygen wasn't there, we would ruin this gradient. And so if there's low oxygen, there's not going to be enough energy to drive ATP synthase in order to charge up the ATP. So we're stuck with what's going on during anaerobic respiration. I think it's a misconception to think about anaerobic respiration as what happens when there's no oxygen. If you have no oxygen, you're dead. Okay? But this is our baseline. Through these whole processes down here, whether we started with that one pyruvate or one acetyl-CoA, we can turn either of those, so pyruvate, or the acetyl-CoA, we can take either of those and use that to charge up 17 ATP. So times 2 equals 34 ATP. So now if we add the 2 ATP from glycolysis to those 34, bam, there's our 36 ATP that we can charge up during aerobic respiration.